So we're here at ISNTD Coinfections and Comorbidities here in London. I have here with me Dr. Lisette van Lieshout from the Leiden University Medical Center. I just wanted to kind of start just a little bit of an interview with you. We hear again and again that the whole issue of diagnostics, the fourth quadrant of the healthcare model, is just not engaged enough in terms of NTDs. It seems to be a huge gap for the programs going towards elimination and eradication. You're here with quite a cutting edge poster in terms of a multiplex diagnostic approach to studying distribution of intestinal parasites. Would you like to just elaborate a little in terms of the poster and your work?
Thank you. Yeah, we used to use a lot of microscopy for diagnostics of intestinal parasites, in particular intestinal helminths. And of course that's a very suitable technique if you have high intensity of infection and high prevalence. But now we are approaching indeed more and more a situation of elimination, then intensity drops. And then suddenly it becomes very clear that microscopy is not sensitive enough and you need more advanced techniques. And one of these techniques is the detection of parasite DNA in a stool sample. And one of the advantages of the multiplex real-time PCR we developed at the LUMC is that you can target for multiple parasites at the same time. So this opens up also data of parasites which have been highly neglected in the past, like Strongyloides, for example. And we suddenly see whole new patterns of this parasite as well. And also what has been mentioned this morning during the conference, protozoa like Giardia or Microsporidium, they're suddenly now detected by using this technique. So it's very helpful, it's very sensitive, very specific to diagnose multiple infections at the same time, just in a single stool sample.
That's fascinating, and it's very interesting to hear that your research has come about, or partly come about, as we approach the elimination tail. Because I've heard from other sources before that as we approach that tail, at least in lymphatic filariasis as an example, the unit cost per treatment, per patient rises dramatically as we approach, smaller number of cases to go out there and find them. So it's very interesting that that's become a driver for this type of technology, as it were. You mentioned this morning, I see a figure there where you're in the middle of on the left to the middle, in terms of the different countries that you've been doing tests in, and you mentioned the protozoan infections with schisto this morning, and they were, I believe, in Uganda,
yes.
Right, in the lakes of Uganda. Which areas geographically have you covered with your work, and what have you seen?
Well, the way we work is that we always collaborate with other groups and they approach us because they have a diagnostic request. And these are studies done in South America, in several African countries, in Indonesia, in Asia. And they're very different, sometimes because groups are interested in immunological aspects or they indeed want to monitor control and intervention. But that's the reason why we have access to so many different countries or different project areas where we can do our diagnostic research. And what we notice is that there is a huge variation in the distribution of the different parasitic infections. So, for example, we were approached by a group working in Senegal in collaboration with the Tropical Institute in Antwerp. And this is a schistosoma project, and they noticed a lot of schistosoma in microscopy, but hardly any soil-transmitted helminths. So they were wondering, could we use the PCR also as a quality control for our microscopists? And indeed, we could confirm that there were no soil-transmitted helminths in this community present. So that again shows that this real-time PCR is a very powerful technique.
Yeah, even if it's to validate existing technology.
Yes, you could even, because the disadvantage, of course, of PCR is that it's relatively expensive and it needs a well-equipped laboratory. So it's certainly not field applicable, but you can imagine that you use the real-time PCR as a quality control of your microscopists who are working in the field.
Yeah. Okay, that's very interesting to augment what's going on. In terms of, you mentioned people come to you with their diagnostic needs.
Yeah.
And I'm asking that for a reason that here in London you've got places like the International Diagnostic Center at the LHSTM with Professor Michael Miles and Professor Rosanna Peeling, which I'm sure you know personally.
Yeah, absolutely.
These people. What would be the difference between somewhere like that and somewhere like Leiden? And I'm asking that for almost a commercial reason, because we try to push progress through partnership. So why would they come to you? What are you saying to them that they're going to come to you? What's the USP? What's the
Yes, well, in relation to the real-time PCR, we were the first who developed it and who published it, and that I think we were well known for that. And we had the settings, we had an automated system also. So we were the first group who did large-scale screening also with the PCR. So that's why in the beginning a lot of people came to us. I see now more and more, really the last few years, very abundantly, that groups copy our PCR and introduce it in their own labs. And right they are. It's very easy, it's published all, and so it can be done. Another thing is that beside PCR work, we also do antigen detection work, specifically for schistosoma. This is based on the monoclonal antibody, and only we have access to this monoclonal antibody because we developed it. So that's another test, but then it's only detection of one single parasite, so it's different, principally different from the real-time PCR. But then people come to us because we have this test available. Mm-hmm. So can use it.
Going back to the dynamics of the test as an example, and moving away from the elimination tail that we were in, you've seen the rise of hybridized schistosomes, right? Could be down to differential reactions to praziquantel. Could be, a whole host of issues that are giving rise to documented evidence where you've got hybridized schistosomes in different species, right? Given that that's going on, what type of impact do you think that's going to have on the take-up of a DNA-based? Your approach.
Yeah, there's more to say about it. The PCR we developed for schistosoma detects all hematobium and Mansonite species, so it does not differentiate. So we have no problems also with different hematobium, let's say, substrains. There is another approach, but that's done here at the National History Museum, where they look at these genetically different strains. So it's just a different. For us, it's important to detect as many schistosoma as possible, without differentiation of specific of species or subspecies. Well, we have also this circulating antigen. It can detect even all the Japonicum species and all the animal species as well. But I think you have to use it then in combination with the technique of the National History Museum to differentiate which actual species or subspecies we're dealing with. Exactly. I must say, if I can add something else about the PCR, is that you can use it not only for stool specimens, but also for urine, and more interestingly, also for example, vaginal lavage or sperm samples, which is very, very powerful for the diagnosis of genital schistosomiasis, which is a very
huge issue,
absolutely, and their microscopy won't help.
It has
in
to
the
be
diagnostics.
this type of technique.
Absolutely,
But that's very interesting. So obviously today, Lisette is all about progress through partnership, and you see quite a lot of diverse groups in terms of the speakers and the participants that are here. From a partnership perspective, I know you're developing an ultrasensitive diagnosis of schisto by CAA detection. Would you like to just elaborate a little bit on that and just highlight some of the touch points for potential partnerships?
Yes, well, at the LUMC we have a long research history on these circulating antigens. Actually, there are two, the CCA and the CAA. And with the CAA, we now develop this ultra-sensitive test using a very, very sensitive label, fluorescent labels. And these tests are based on the use of a monoclonal antibody. It's now a strip-based, a lateral flow assay. And we really, searching for partnership to make this test available also in a broader sense for other communities and for research purpose.
Okay, well, we're going to flash your email up underneath on this. So if anyone's out there looking to make partnership in terms of this particular area, be sure to contact Lisette directly by email, and it's there. So we heard from the morning session a little bit about the immunological landscaping in terms of the nuances between host and parasite interactions, and it was a kind of a generally accepted norm or fact from this morning that there is not enough basic research involved at the host pathogen level in terms of understanding just look at the leprosy case that was covered this morning with deworming and giving 2 different types of immune response, which could then go on to give you a leprosy or non-leprous situation. And I know Russell Stothard had touched on it on his schistone and giardia as well. In terms of the immunological landscaping or the better understanding of the interactions between host and pathogen, what type of impact could your tool have on that understanding?
Well, actually, within our group, within the Department of Parasitology at the LUMC, we do a lot of research on the interaction between helminths, schistosoma, hookworm, and other pathogens, or even the non-communicable diseases. And again, there very sensitive and specific diagnostic tools are essential if you want to study this interaction, the immunological interaction, the pathological interaction between the parasites and these non-communicable diseases or these other pathogens.
Okay, so you're impacting that quite heavily, actually, by the sounds of it.
Yes.
Well, thank you very, very much for making time for a short interview. I hope you're enjoying the day, and I know the afternoon session is about to start, so thank you very much.
Thank you very much, and I indeed enjoyed the symposium a lot. So thanks.
Thank you very much.